My title is Improved Liberibacter Detection in Plants and Insects. And so I'm actually going to do this just in this room. If anybody out there is not a molecular guy and you're collecting samples, one of the main things that you can do to help us is to make sure that you freeze it. If you're collecting insects, if you're collecting plant matter, if we're doing molecular techniques on it, get it cold as fast as possible. And then that's the end of my talk, right? One of the things that, thank you. One of the things that we always have to come up with is ways to work around sample preparation issues. And I understand the issues. If you're out in the field and you collect things, it's not always easy to go ahead and throw it in a minus 80. But the colder you can get things, the faster you collect them, the better off we are downstream to do what we do with our stuff. Because when stuff dies, it starts to produce DNases and RNases, and that impacts our ability to do a lot of the things that you heard about earlier in this section. So get it cold. If you're just collecting, you see something odd, always put it in the freezer if you're going to give it to somebody to assay. So just get it cold and we'll be good. So I wanted to start, besides that, just talking about some of the things that we've been doing going the other direction from Texas, going south. And I showed this yesterday, which is kind of some work that I got involved with Joe on, where we were looking for pathogens in different crops, or Liberibacter in different crops throughout Central America. And as we were moving down through Central America, we were working with people at Zamorano University and asking them to just look in solanaceous crops, and if they see strange symptoms that don't fit with traditional virus or phytoplasma problems that they've seen, to collect it and let us take a look, and also collect insects off these plants. And most of this worked pretty well. We went down and did some transfer of knowledge where we were showing the things that we were finding in the U.S. And with that group, it was really good because they had an outreach program, kind of extension-based type program, where they were going to be out in the fields and they were going to be able to look for some of these things. And that's where we were teaching kind of this idea behind recognition in different solanaceous crops, proper collection techniques, make sure you get it cold, storage, also some vector identification so that we could get good samples. And as I move forward with this, and you've heard a lot of molecular stuff today, I want you to understand that most of us that work in a molecular lab also understand that not every technique is right for everything that we do. Sometimes traditional PCR is a really good technique, especially if somebody doesn't have access to real-time PCR. And so as diagnostics go, we can do a lot of highly molecular things, but it doesn't mean much to large-scale programs that need data fast. And so there is appropriateness for these different techniques as we go through. Estella actually had real-time PCR in her lab and then moved to a different organization, and she no longer had real-time PCR, so we changed the way that we were doing some of our techniques. So this is a picture of tobacco, and this is a paper that came out last year where some strange symptoms were recognized in tobacco, and they decided to sample those plants. They sent DNA legally to the United States through Joe's lab. And we were able to confirm that it was PCR positive for Liberibacter. They had found potato psyllids on tobacco. Those were identified as potato psyllids. And we did a pretty good job. So that was an example of the system working really well, where we had good field work, good sample preparation. It was DNA that was prepped in Honduras, and then it was shipped properly to the U.S., and we got really good sequence out of it. And if you're going to do the publication part of it, that's important, not just for us academic people who need that for advancement, but also it offers us the opportunity to get the information out to the general public. And it's also vetted and important. And plant disease required sequence data from multiple genes as confirmation of these things being there. So there was quite a bit that had to be done at the front end, quite a bit at the back end. So we used everything correctly, and we used a mixture of traditional PCR, qPCR, and then also sequencing to confirm what our PCR positives were. And yet, technology keeps moving forward and offering us sort of that next step. In most molecular labs right now, you have access to real-time PCR. So that's becoming kind of the standard where PCR was 10 years ago, just traditional PCR, thermocyclers. And so kind of looking for that next step in the technology that's going to help us. And a few years ago, 5 or 6 years ago, I started looking at kind of a metagenomic analysis of potato psyllids. And so we did 454 pyrosequencing. This is done with titanium plates, so it's expensive. Just the materials are expensive. Now we have things like ion torrent and Myseq, which uses a lot more plastic, so assays become much less expensive. And the more people, more researchers that are doing this, the cost starts to come down, kind of following Moore's law. And so we had this kind of unique opportunity to use 16S markers to see all of the bacteria that were in samples. And so a couple of years ago, I did a pretty simple analysis where we took 25 samples of psyllids from California, from different places that were provided by John Trumbull. And then we also took samples from the central U.S., collected from everywhere from Texas to Nebraska. I think we had some Kansas in there and multiple places in Texas. And this doesn't show up very well, but you'll see green blocks in here, and you'll see black blocks. Where you see black, that means that there weren't any samples, so those would be zeros. Green was a low copy number, and the brighter red it gets, the more copies there are. So these are the bacteria that were contained within these insects. And I was interested in this from a phylogenetic standpoint because I thought this would be a good way to differentiate, and it was. We had good branching. We had good branching up here, which was based on location. Everything from Texas in the middle of the U.S. kind of fell together. Everything from California fell together, so it made sense. But the cool thing that I want you to take a look at, and this came up this morning in one of Joe's talks, was the Wolbachia that were present, and then also things like Carcinella rudii. And these are symbionts. So the things you see in high copy number up here in mostly all of the psyllids, these are endosymbionts. So these are things that are pretty tightly related to the psyllid. As you go down this line, things that are less abundant are things that are probably transient, things that live in their guts and maybe contribute to some kind of life history. But it makes a really interesting way to look at this. And we did find Liberibacter in a lot of samples. In some samples, we had super hot psyllids that were out there. And these are all field-collected psyllids. So that's interesting. I'll get to that in a second. But we were able to use this, put phylogenetic trees together, found out that there were some California psyllids that showed up in Texas and some central psyllids that ended up in California. So we had that kind of overlapping range. And so we continued working with this data, and we collected this library of information over the past couple of years. We use pretty straightforward bioinformatic tools to go through this. And this is what an average psyllid comes out when we put it onto a coronagraph. And because our sequencing gives some variable results as whether or not we can call things a certain genus or species, we kind of have to look at it in a layered look. But you can see a typical psyllid usually has quite a bit of Wolbachia. So I'm really interested to find out about your no Wolbachia insects. I think that's really cool, especially with the next thing I'm going to show you. But we usually have pretty low numbers of Liberibacter that we're finding, but then we have other things like our other symbionts that show up in high numbers. And so after I heard what you guys were talking about, I went back up to my room and kind of did a quick data analysis on some other part of this, which is kind of comparing our Wolbachia finds to our Liberibacter finds, and also comparing those to Carcinella, because that's something that shows up in quite an abundant way. And what this basically tells you in kind of an ugly way, I did it quickly, is that if you have Carcinella rudii in high abundance, it limits the number of Wolbachia or Liberibacter you're going to have. And that works for all of them. So if one of them is way out of whack, the other ones are going to be in smaller numbers. And so the no Wolbachia thing might lead to the idea that with less Wolbachia, you might have more Liberibacter. So that's an interesting finding in those populations. So we went on from there, and we took another large data set with 127 samples, had carrot psyllids as an outgroup, looked at the bacteria, put a PCA together, most things grouped together. Carrot psyllids were kind of different than potato psyllids, and potato psyllids from different areas of the United States kind of grouped together, where we had what I would call our northeast or northwestern haplotype, and they kind of grouped away. But what I really wanted to get to was the next analysis that we did kind of along this way. Took another 110 psyllids, had 100 field collected psyllids, 10 lab colony psyllids. We did traditional diagnostics with traditional PCR with multiple primer sets, and then also the WinBin real-time PCR assay, which is the APHIS standard, and then did 454 pyrosequencing on it. And the results were kind of striking. We found that if a psyllid had a lot of Liberibacter, then it tested positive using PCR. But if we had psyllids that, with our pyrosequencing, which is kind of like an absolute sequencing technique, if they had fewer than 300 reads, they would come up negative by real-time PCR. So I think when we're doing PCR in the field, we're missing some Liberibacter that's out there. We're getting some false negatives. And I don't know the biological significance of that, because it's possible that level of Liberibacter is not transmissible, and then from a point of view of field entomology, they might not be super important. But I think that gives the idea that there's some Liberibacter out there that we're not recognizing as kind of sitting there in really low copies. But if we had super positive samples, then we found quite a few copies of this, which makes sense. But you can see the difference out of that 110 samples. We had 21 positives using the 454, and we only had 5 positives using any of the 3 PCR techniques. So it was a lot more sensitive. In this, in our lab-reared colonies, turned out that an average of 73% of the bacterial reads we got back were Liberibacter. So that means that 3/4 of all of the bacteria that we're finding in there was Liberibacter. And these are colony insects that are on infected plants in a hot colony. This was a little different when we looked at those that were in our field-collected samples that had about 4%. So what we're seeing out in the field is a little different than what we have in our lab colonies. So I think we always need to keep that in mind when we're doing lab experiments and we're coming up with large ideas of what's happening in the field. That just summarizes most of those ideas. So now we're transitioning to Myseq. It's a little bit less expensive platform. We get a lot more bang for our buck. With 454, it was pretty expensive, $150 a sample. So I'm thinking if Jim Crosland was going to do that with his system, his budget would have been a little bit higher and probably not fundable. But and we would only get about 6,000 sequence reads from each sample. Now that we moved to Myseq, right off the bat, we went to $80 a sample and got 35,000 sequences. And we've been working our way to cut that in half. So now we're at 40,000, or 40,000. We're at $40 a sample, and we're getting an average of 15,000 reads. And we're still getting pretty good clarity, much better than we even were with 454 for a lot less money. And I think our goal is to bring this money down, or bring the cost down to $10 per sample, which I think will be competitive with qPCR. So I'm thinking this is the next sort of step in the evolution of these molecular diagnostic techniques. And so just to show you some last little piece of data, we ran through, we were trying a couple of different ways, and we had an interesting conversation with Don Henney at the Southwestern branch last February, and he had tested some thrips for Liberibacter, and he found it, and I think he did transmission experiments. I can't remember exactly how that all worked out, right?
No, not exactly transmission.
But they were, yeah, and they were testing positive. So we wanted to follow that up because our lab colony grew thrips. So we wanted to look, and what we found was that in our psyllids, we had fairly high copy numbers in 2 of our samples. 43%, 20% of the reads came back Liberibacter. We had 1 which wasn't terribly hot, but we kind of backed up the data that you guys found, which was the thrips did test positive, although relatively low copy numbers. So the significance of that, we're not really sure, but in light of some of the other things that we've seen in other solanaceous crops, this is an interesting thing to think about as we kind of ponder the entire system. So just to end, we have colony insects. They're not always representative of what wild psyllids are doing. I think we'll get down to $10 a sample and still have between 5,000 and 10,000 reads, which will be similar to 454. The downside, this isn't fast, so it's not like running PCR in your lab unless you have the money to buy a Myseq yourself. And so usually it takes a week or so to get some data back. And we also want to do some transmission experiments to QC this methodology and also make sure that this is bringing us the same results in plants. So I want to start to find, somebody asked also about how you get from haplotype to biotype. And so documenting the changes in their biological behavior, sensitivity to insecticide, transmission rates, things like that, correlated with that genetic background is what's going to do it. So thank you guys for listening. I think we have one more talk, and then we get to have a discussion. Thank, of course, the funding agencies. I forgot to do that on my first talk, but the SCRI and the mini grant program definitely. So thank you.